Immune to SARS-CoV-2, finally, thanks to an emergency vaccination. That is exactly what large parts of the population and the government want right now. Pharmaceutical companies are being given considerable support to advance the goal of a corona vaccination as quickly as possible. But how quickly is ethically justifiable? How many risks are being hidden because of the speed of development? Professor Stefan Hockertz, a biologist, pharmacologist and toxicologist, points out a number of unanswered questions and warns that millions of people will intentionally submit themselves to bodily harm if these questions are not answered ahead of time.
Schönen guten Tag erstmal, Frau Schmidt. Ich bin seit 30 Jahren als Toxikologe und als Immunologe. Ich habe ursprünglich mal die Immunologie gelernt und bin dann in die Toxikologie gegangen.
First, let me say hello, Ms. Schmidt. I have been working as a toxicologist and immunologist for 30 years now. Originally, I studied immunology and then I went into toxicology. I have been working in drug development for 30 years, and that includes 1 year in vaccine development. This means that I advise companies that are active in the field of vaccines and develop new vaccines there, but I also work very closely with the Paul-Ehrlich Institute, which is responsible for vaccine approval in Germany. So I have a pretty good regulatory background in the field of vaccines and have been active in that field for 30 years.
Very good. We could hardly find a more qualified expert. Can you explain to us in a simple way how the current approach to vaccination can be described?
Ja, Impfungen sind, und das möchte ich mal vorherschicken, aus meiner Sicht eine der größten medizinischen Errungenschaften.
Yes, vaccines are, and I would like to say this in advance, in my opinion, one of the greatest medical achievements we have in modern times. They have saved us from many, many diseases. We have been able to eradicate these diseases to the greatest extent possible, such as smallpox, by convincing many people to get vaccinated. Seen in this light, my basic attitude towards vaccinations is positive. Until now, vaccines have been killed or attenuated bacteria or viruses. Killed viruses or attenuated bacteria or viruses, I now call them exciters, which are designed to infect a person. In the case of smallpox, which I just mentioned, it was cowpox, in other words, a pathogen or exciter that was attenuated, as we say, meaning attenuated using an animal. In the case of influenza, it is an influenza virus that is attracted via the hen's egg and then attenuated, and then infects us. We become infected with an attenuated pathogen during a vaccination so that our immune system can learn against it without danger in order to be able to protect us quickly and actively if we become actually infected. Up to now, we have had to deal with continuously altered, attenuated pathogens that we and our immune system could cope with relatively well. This is the principle of the vaccination we have been following for many years and have been doing it successfully.
Wie wir sie seit vielen Jahren durchführen, auch erfolgreich durchführen.
For the SARS-CoV-2 vaccination, the pharmaceutical industry is now taking a new approach because the vaccine is supposed to be developed in a very short time. What effect will the new vaccine have on humans, assuming it does work?
Ich muss vorherschicken, dass die Herstellung eines Impfstoffes sehr schwierig sein kann.
I must say in advance that the production of a vaccine can be very difficult. You will have noticed, if we look specifically at influenza, that the influenza vaccine from the year 2020, which you can get now, is produced using the information approximately from 2018, at the latest 2019. This is not because the researchers do not know what the 2020 influenza pathogen looks like, but because it is so difficult and it takes so long to produce the vaccine. I just mentioned that it is sent through the hen's egg. That's where it has to be grown. So it's a very, very lengthy and costly, especially lengthy, production process, which takes 1 to 2 years for just the production alone. This is how, some time ago, by the way, an alternative method was found. The German company CureVac has been working on this topic for 12 years without having obtained a successful approval so far, namely, a system that only imitates the genetic information of a virus. And this genetic information, in this case it is supposed to be a messenger RNA or single-stranded RNA, is transported into the cell via a vector system or carrier, as if I put it on a little truck to be carried in there. This is a completely new approach, and this is very important information. There is not a single vaccine based on this technology that has been approved worldwide. So there is not a single vaccine based on viral mRNA or viral DNA that is transported into a human cell via a vector to be read. This is a completely new vaccine, and surely one for which it is worth doing research. There is no question about that, because it is relatively easy to produce genetic material. It is simple and it is fast. And now I speak as a toxicologist. The question that arises is, if I want to develop such a new vaccination strategy, and this would be one, then I need to know very precisely about this strategy. I need to know about the distribution of this vaccine in the body, about its function or pharmacodynamics, and especially about any side effects that may occur. And these are many and varied when I transport genetic material from a virus into a human cell. Ultimately, this can be done via enzymes. There is, for example, an RNA-dependent RNA polymerase that is able to multiply these ribonucleic acids, which I have now had transported virally from the laboratory bench into my cell. That is possible, and this would then lead us to actually causing a change in the human genomic structure. And that is what needs to be tested. And now we have the situation where this new vaccination strategy that we have been informed about for about 3 months regarding SARS-CoV-2, in about this time, in these 3 months, development work has been done, which has, so we are told, already got all these risks under control and eliminated them. Because the Paul Ehrlich Institute, together with an ethics committee of the University of Tübingen, has allowed that 168 people are already to be treated with this vaccine or on an experimental basis. I do not consider this to be right, and I find it ethically unacceptable, simply because the necessary data on the safety of this vaccine could not be collected in these 3 months.
Weil man schlicht in diesen 3 Monaten die notwendigen Daten zur Sicherheit dieses Impfstoffes nicht hat erheben können.
I was about to ask whether ethic advisory boards come into the picture, not only from the University of Tübingen, but from the Federal Republic of Germany and worldwide, in order to discuss what is essentially genetic manipulation. In Germany, we are still not even very liberal when it comes to genetic manipulation of plants and animals. Or is it not appropriate to speak of genetic manipulation here?
Ich stehe, wenn ich auf diese Frage eingehen darf, vor einem ungeheuren Rätsel, und es stellt letztendlich auch unser aller Arbeit im Bereich der Regulatory Affairs, der Zulassung
If I may address this question, I am faced with a tremendous mystery, and it ultimately completely challenges all our work in the area of regulatory affairs and the approval of vaccines. Yes, the question arises, under what ethical aspect has this clinical trial been approved at all? And I know of a statement by a member of the Tübingen Ethics Commission who actually only said, well, when I eat a steak, I take genetic material from the cow. That doesn't hurt me either.
Oh.
There is, and I may say this now without emotion, a difference between whether I take genetic material orally, digested in my stomach, and then excreted again, or whether I get it administered intramuscularly with an artificial transport system so that it can enter my cell directly, protected, and be read off there. I say this so clearly because I have the feeling that common sense has been exposed in many people who have also had to decide which clinical trials are to be conducted on a SARS-CoV-2 vaccine. I've asked the Paul Ehrlich Institute, as I keep asking different institutions, because I want to understand scientifically what is happening there. I want to hear arguments, and I have asked the Paul Ehrlich Institute what happened in these 3 months, which from all our experience as toxicologists and pharmacologists and also immunologists will certainly take 5 to 6 years. What has been achieved there in 3 months, which otherwise can only be achieved in 5 years, in order to guarantee the safety of a vaccine, a completely new vaccination strategy of which we know far too little? And I have not received an answer so far. And I have added to this question, and perhaps this is a good answer to your question as well, I am of the opinion that under no circumstances should we sacrifice our science, our ethics, and our regulatory guidelines for the political declarations of intent from ministers and a chancellor. And that is exactly what is happening.
But we know now that testing is going on now. We know that people have already been given a vaccination that seems to work according to this new method. Are you already aware of the results, or is all this still much too new?
Da ist die Frage, was ist ein Ergebnis?
There is the question, what is a result? On the one hand, of course, we are looking to see whether this vaccination, in other words, the transport of viral DNA into our own cells, causes a change in our genetic structure, our reading device, up to the possibility that this mRNA will become independent. The rationale behind this is that these cells, our cells, are supposed to produce viral material in order to be recognized by our immune system. The original problem would be that we can basically only measure antibodies, and that would be proof that this vaccination works. However, we have an immune system that can have many different modes of action, and one mode of action is, for example, cytotoxic T cells, or killer cells, which now recognize and kill our virus-infected cells. And these killer cells are very difficult to detect. So, even in the rationale of the approach, when we work with viral DNA, we have a problem to show whether this vaccination really works. It works when I detect antibodies. But if I don't detect antibodies, it doesn't mean that it hasn't worked. It could also be that killer cells have been activated. This is a general problem that we know repeatedly from other vaccinations. When we vaccinate against hepatitis, we always have a number of patients in whom we cannot detect antibodies, even though they have protection. But that is not the danger behind it. I would like to talk about the dangers that have not been looked at. So, if experiments are now being conducted on humans, in this case 168 healthy volunteers, you have to remember that we are dealing with healthy people who are given something to prevent them from becoming ill in the future, that is the intention of a vaccination. This means that our requirements for this vaccine must be much higher than, for example, requirements for therapeutic treatment. With therapeutic treatment, the person is already ill and can die in case of doubt if I do not treat him. So I can accept greater side effects or greater problems as part of the risk assessment. But when I do a risk assessment for a vaccine, it should not harm the person as much as possible because I am giving it to a healthy person who can only possibly get sick. The risk assessment of a vaccine must be much higher. What is the risk of this vaccine if I give it to people? The transport system I just mentioned is contaminated. Contaminated in the pharmacological sense means that it is 99% pure. Even if I only have 1% impurity, such a preparation is already dirty.
What does that mean exactly? Where does the contamination come from? Is this the active material, or what is the impurity?
Das Wirkmaterial, das heißt also, die RNA ist relativ sauber herzustellen.
The active material, in other words, the RNA, can be produced relatively cleanly. However, it must be protected because our body has very active systems to kill this material extracellularly or to destroy it. This also makes sense because the body wants to protect itself against free RNA being present in the body. For example, if cells were to break down, RNA would also be released. Therefore, the RNA that is now produced in the laboratory has to be packaged in a carrier system. And these are nanosomes or liposomes. These are lipids, these are envelopes, and these are to be regarded as an impurity per se because they are a type of dosage form. And from our experience, these nanosomes and liposomes are very difficult to produce in high purity. Why does the whole thing have to be highly pure? Well, because we do not have a so-called no effect level in the immune system. This means that we have no area in the immune system where we can say that the immune system does not recognize this, that it's subliminal. This means that we have to look very carefully at what we offer the immune system together with the genetic material. That takes time. That requires toxicological expertise, and in my opinion, this has not been done so far due to time constraints.
Und das ist nach meinem Dafürhalten bisher schon aus Zeitgründen nicht geschehen.
That is just one of the problems. But Now I would like to summarize this in very simple words, and please tell me if I have understood it correctly. This RNA vaccine is transported into the cells, then these cells change in such a way that they are mainly recognized by our immune system, so that action is taken against them. But now these are the body's own cells. How can I know how far they will reproduce?
Das ist unbekannt. Das ist ja genau eine Frage, die ich stelle.
This is unknown. That is exactly the question I am asking, to what extent we actually have this genetic material from the virus under control. Basically, we do nothing else but imitate a virus infection, but by transporting a gene snippet, artifactually we call it, a gene snippet into the cell. What exactly happens with this gene snippet? We do not know. We have hopes there. We have the hope that this gene snippet will be read by the ribosomes and that proteins will be produced and shown on the surface of the cell again, thus creating the illusion of a virus-infected cell. Afterwards, this gene snippet should be switched off and not reappear. This is our hope, but it has not been proven. That is why the questions we ask are allowed. What happens to the genetic material that I transport into a human cell with an artificial transport system? And for the first time. This has never been done before. What happens to the material? Is it duplicated? It is biologically possible for it to multiply. Is it incorporated into a plasmid? That is also theoretically possible.
What does that mean?
This means that it is then present outside the nucleus as a gene snippet, and yes, it is passed on as well. Do such gene snippets only get into the muscle cells, or are they so well protected that they may also enter the germline, meaning the egg cells of the woman and the sperm cells of the man? Then this gene snippet would even be passed on to our offspring. None of this has been clarified. All I am asking for is that this be done in lengthy, elaborate investigations of which I am aware. But when I hear from our politicians that by the end of this year we will have this vaccine available for 80 million people, then a horror scenario opens up to me where I say, all these questions have not been answered and could not be answered at all because we've only been working on this topic for 3 or 4 months.
Konnten gar nicht beantwortet werden, weil wir erst seit 3 Monaten an diesem Thema dran sitzen können oder 4 Monate.
Mm-hmm. You yourself are involved in the testing of drugs, so I would like to know the normal process of such a development, that is before corona. You can certainly tell me how large must the number of tested persons be? Will a double-blind study be conducted at the highest current scientific standard? And then, how long do you have to test to determine the effects and side effects? And when and how are they able to be discovered at all?
Ja, jede Impfstoffentwicklung, aber jede Entwicklung eines Arzneimittels beginnt mit der präklinischen Entwicklung, also all dem, was vor dem Menschen stattfindet, die Sicherheit.
Every vaccine development and every drug development starts with preclinical development, meaning everything that takes place before testing on humans to ensure the safety of such a product before it even comes into contact with humans. These are studies mandated by regulations, conducted on cell cultures, then also on animals, in order to show early results. How is the vaccine distributed in the body? At first, we don't even look for effects. We are only looking at safety. In a second aspect, we also look at the effect. Animals are also vaccinated accordingly, and then we look at the effects. Which test system can I use to prove the success of this vaccination? Which test system can I use to prove the effect of the vaccination? These preclinical studies take about 3 to 4 years for a new active principle, depending on how rigorously these studies are conducted and how often they have to be repeated. Only then, usually after 3 to 4 years, do we receive approval for a first experiment to be conducted on humans. This is what we call a phase 1 trial, and it is usually set up relatively small in order to keep the risk to humans relatively low. There are usually only 20 to 30 subjects, not 160 as there are now. These 20 to 30 subjects get the vaccine, but they are only briefly screened and looked at. What is my acute difficulty? Do I develop fever or other side effects? I can't even look at long-term effects in this phase 1 study, but I am looking at what acute side effects can occur in this area. If these are already too great, that is the end of this vaccine. If they are acceptable, such as maybe only reddening of the skin at the injection site, then I can continue for the time being, but of course still with a high risk. That's why we will go then into a so-called phase 2 and also phase 3 clinical trial. The phase 3 clinical trial is then the blind or double-blind study you mentioned. That is always provided with a placebo, where neither subject nor physician know which subject received the actual vaccine and which not. This should protect us from interpreting too much into data to meet our expectations, but that we can actually evaluate the effectiveness, but also the safety of this vaccine completely free of bias, only statistically. A follow-up period for these vaccines is usually 2 years. That is approximately the time in which one can see whether, in fact, also long-term damages can occur, so-called vaccination damages. This follow-up period is usually set at 2 years to show that one is really on the safe side, that one does not harm the person, and that the vaccination is only useful to them. This means if you add up all the time, phase 1, 2, and 3 studies, double-blind, together with the preclinical phase, we would have a time horizon of about 8 years, after which we can actually make the vaccine available to people in good conscience from the first preclinical study to the approval of the vaccine.
Den Menschen tatsächlich auch guten Gewissens zur Verfügung stellen kann.
Are there any reviews of pharmaceutical studies, like other scientific studies or scientific publications, so that others can also review these developments and scientific information?
Das ist wünschenswert.
It is desirable in the field of science to always mirror the data we have with other scientific data, and that the whole thing takes place in a peer-reviewed context. This is sometimes difficult to do for reasons of secrecy because I may be in competition with others, but in vaccine development this is basically common practice because we naturally want and need to make use of the broad knowledge of science to ensure the safety of people.
So in this case, not really?
In the case of corona, there is no diversity of opinion anyway. We've had to experience this painfully in the last 4 months, in that opinions are not only not heard, but also suppressed. I may now quote as the last very current example a podcast of Markus Langemann at Langemann Media, which I led 3 days ago, and which was forbidden today by YouTube and was deleted. That is not diversity of opinion.
Now, another question, because you spoke earlier about the competition in the pharmaceutical industry. Where is the most advanced development currently? Who is already testing? And will the others stop right now because it's already too late anyway? Or how does that work in the normal pharmaceutical business?
Der Markt, besonders für die SARS-CoV-2-Impfung, ist so riesengroß, dass das ein Unternehmen gar nicht gewährleisten kann. Es würde sich hoffnungslos übernehmen.
The market, especially for the SARS-CoV-2 vaccination, is so huge that a single company cannot even guarantee it. That would be hopelessly overkill. Look, the Federal Ministry of the Interior and Mr. Spahn have already announced that they have already ordered 80 million doses of a vaccine that has not yet been approved. It has to be produced, it has to be properly packaged in accordance with good manufacturing practice, it has to be safe and proper and always traceable. I know from the company Moderna that they are already conducting clinical studies on test subjects. I'll leave out the word voluntary test subjects, they are just test subjects. I'm not sure where they come from. There's a lot of literature that says they're supposed to be soldiers, and the voluntary aspect is perhaps not quite so far off. In Germany, it is the company CureVac and the company BioNTech who have been working on this issue for many years, and that's perfectly okay to gain market share here by making this vaccine available early on. But with the comprehensive necessity as conveyed by governments that are even considering compulsory vaccinations, the market is so huge that there is more room than for just one company. This means that even if Moderna starts the clinical trials now and can show first successes, other companies will work on this topic and follow suit because the market is basically still big enough for many, many companies. In particular, because there could be a compulsory decree that 80 million people or 83 million people in Germany are to be vaccinated. The issue is not off the table. And of course, this involves particularly high risks, if I may say so. If I vaccinate only a few people and accept a percentage of vaccination damage, then that still requires a very cautious risk assessment. But you can make and estimate such a risk assessment and then think about whether it is ethically justifiable or not. I am often asked now, where do the numbers actually come from? I follow the figures given by a Mr. Gates, whom I am reluctant to quote, but in that case I do at times. If we assume that even only 5%, and this is a relatively small number of people who are vaccinated, show vaccination damage, then in 80 million people, this means 4 million people who show vaccination damage. If I am responsible for this vaccination damage because I made the first examinations far too short, then that is 4 million people who were intentionally injured. I can, of course, reduce vaccination damage or reduce the risk of vaccination damage if I investigate this vaccine long enough and reasonably enough with state-of-the-art technology according to the regulatory requirements. If I do not do this, then there is intent involved. Then it is intentional bodily harm. If I'm talking about a mortality rate of 0.1% for 80 million people, and these are also figures from the same source, then I am responsible for 80,000 deaths from the vaccination. If I vaccinate 80 million people, and that is a figure we cannot really imagine, 80,000 deaths. Perhaps this can be imagined by saying, this is the city of Constance or the city of Bamberg that will be wiped out.
Is this the normal level of mortality due to a vaccination?
Das ist schwer zu sagen, das ist von Impfung zu Impfung unterschiedlich.
This is difficult to say. It varies from vaccination to vaccination. For example, when we talk about pneumococcal or meningococcal vaccinations, meningococcal vaccinations are better. I have an increased mortality rate, especially when people have to fight another infection at the same time. This is always a big problem with compulsory vaccinations anyway, because I have to take a medical history of each individual case. Does this person currently have an infection? Does another person have difficulties with the immune system? Because a vaccination is always a strain on the immune system. If we look at the meningococcal vaccination, it is certainly risky. Again, we have mortality rates that should be far below 1%, that is 0.001%, but it depends on the anamnesis of the person. A sad example of how high mortality rates can be with meningococcal vaccinations we see in connection with corona. In Bergamo, 35,000 people, these are the data from the ISS in Italy, were forcibly vaccinated in January after a meningococcal epidemic in January. That alone was basically a mistake, because this vaccination is of course associated with the flu season, a time when we are constantly coming into contact with viruses, whether influenza viruses or coronaviruses, which both cause respiratory diseases. We should be avoiding this. And every third semester student learns that meningococcal vaccination is risky, especially if there is a risk of contracting another viral infection. This has now obviously happened, and we will see the result in Bergamo, very, very high mortality rates, due to corona, as it has been said. I say with corona, but ultimately caused by this indeed negligent execution of a compulsory vaccination against meningococcus. This means that a basic death rate, risk rate, is actually dependent on the type of vaccination as well as the short and long-term damage that can occur. But we know that we have to be very careful with humans because the vaccination puts a lot of strain on the immune system. And we also have to be very careful with the development of the vaccine. And I'm calling for this care. And this care is obviously not being taken at the moment. The point is, in fact, that we are always trying to get scientific data, scientific statements from the acting and consulting institutions. We are asking, in fact, please give us data, please give us arguments with which we can also reflect our contradicting views. After all, we want to go into scientific discourse, but we get nothing.
But you are normally in permanent contact with the Paul-Ehrlich Institute, aren't you?
Ich bin mit dem Paul-Ehrlich-Institut viel im Austausch. Ich kenne dort auch eine ganze Reihe von Personen. Ich nenne jetzt ganz bewusst keine Namen.
I have a lot of exchange with the Paul-Ehrlich Institute, and I know quite a few people there. I am deliberately not mentioning any names now, but I have been told very, very much by many people. What is happening here is a catastrophe, they tell me. It is a catastrophe how we are influenced. It is a catastrophe how this vaccine is to be approved here. We cannot support this. Then I say, yes, come out with it. Say it, come out. And then there is always the big fear. No, I cannot do that. I'll be out of a job.
We ask ourselves, what is it at the source of these fears? You heard Professor Stefan Hockertz, toxicologist and pharmacologist, From 1986 to 2001, he conducted research at the Fraunhofer Gesellschaft in Hanover. From 2000 to 2001, he was a private lecturer for toxicology and pharmacology at the University of Hamburg. And from 2001 to 2004, he was professor of molecular immunotoxicology. For many years, he was also chairman of the Institute for Experimental and Clinical Pharmacology and Toxicology at the University Hospital Eppendorf in Germany.